Hi, good afternoon everybody. My name is Simon Watley. I'm a member of the IMechE's Water and Wastewater Interest Committee. We're pleased to introduce Dr. Joe Burgess from Isle Utilities, who's going to speak to us today about COVID-19 transmission and epidemiology in wastewater. Joe has 20 years experience in the water sector, having worked in research and development in Britain and South Africa, then as a research manager at the South African Water Research Commission. Her portfolio included drinking water treatment technology, then later the mine water treatment and management program, finally adding sustainable industrial wastewater management. Joe is responsible for South Africa's National Water Research Development and Innovation Roadmap from 2015 to 2025. Her background is research carried out in academic and industrial partnerships. While head of biotechnology at Rhodes University in South Africa and as research officer at Cranfield University in the UK, she involved the employers of biotech graduates in curriculum and fourth-year project design, and her postgraduate students' projects had industrial partners. Joe was the 2014-2016 chair of the board of the Water Institute of South Africa and remains director of the company. She is the previous chair of the Mine Water Division and fellow of the Water Institute of South Africa. She is a member of the Royal Society and the International Water Association and founding president of the International Water Association and the Water Institute of South Africa Young Water Professionals. Joe is an editor for journals Clean Water, Water Science Technology and Environmental Technology Reviews. I'd just like to remind listeners that there's an opportunity to ask questions on the right hand side of your screen. You should find a tab which says ask a question. So if you've got any questions, please type your questions in there, and we will send answers to those as we go along. Thank you very much and enjoy the presentation.
Good day everybody, and thank you for the introduction. As said, my name is Jo Burgess. I work for a small company called Isle Utilities, and you might have met me online already through the Water Action platform. Today, on a similar topic, I'm going to be talking about SARS-CoV-2 and its fate, behaviour, risk and testing in water, but primarily in wastewater. And to start with, just for a little change of pace, I'm going to take us back to basics, and we're going to look at what SARS-CoV-2 actually is. We've heard a lot about it in the last 18 months, so just a bit of microbiology to start us off. So COVID-19 is the name of the disease caused by the most recently discovered coronavirus SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome-Related Coronavirus. I'm going to be using some words including virion, which is the complete and infective viral particle, and the virion consists of the RNA, the blue squiggle that you can see in the diagram on the right-hand side of the slide, surrounded by an envelope, and this is a phospholipid envelope, much the same as many other microorganisms, and you can see here that it's got a number of spike glycoproteins embedded in it, and those spikes are the things that the vaccines recognize and work against, so those are important. The coronaviruses are a group of related viruses that cause lots of diseases in mammals and birds, and they're not new. There are already 4 other endemic human coronaviruses, or HCOVs, plus the famous ones that we've heard about, MERS, the Middle East Respiratory Syndrome, and SARS, the Severe Acute Respiratory Syndrome, that came out in about 2003. They're very closely related, which is great. It means a lot of the measures that we had in place because of previous coronaviruses still hold true for the current one. And they have that absolutely gigantic piece of RNA. That blue squiggle is a beads on a thread conformation. and it's huge. It's about 74,000 kilobase pairs, which might mean something to some of you. And it's very good news because it makes RNA detection much easier than with viruses that have much smaller RNA strands. So this is important to us in the wastewater industry. When people are infected with SARS-CoV-2, they shed both virions, so those whole infected particles, and pieces of RNA from broken virions through their bodily fluids. And this is true whether they have symptoms or not. And this means that wastewater-based epidemiology or environmental monitoring is a very good way of monitoring outbreaks with asymptomatic patients or pre-symptomatic patients. People that are infected with SARS-CoV-2 carry on shedding the RNA and the virions for at least 7 weeks after they recover if they're ill. And this means that we have a very good indication, which is not a clinical indication, of the number of people in the community which are carrying the virus around with them. And that means that wastewater and the receiving water bodies that it flows into, therefore, have a very significant role as a source of epidemiological data. They don't have a very significant role in terms of public health risk, and I'll explain why in a few slides' time. Coronaviruses are not typically waterborne viruses, which is a great thing in terms of infection and transmission, but in terms of working with them or removing them from wastewater, it's a little more difficult because our standard virus detection methods are aimed at waterborne viruses and they don't work very well for the coronaviruses. And the last thing on this slide that's important to note is that the presence of RNA, if we find RNA in wastewater, it doesn't mean that there are infected virions. It doesn't mean that there are whole virus particles that can make you ill also present in the water. So about a year ago, the $64 million question was, is COVID-19 a waterborne virus? Now the short answer, happily, is no. The transmission route for COVID-19 into human beings is through the eyes, nose and mouth. So this is why if you are wearing a mask, it's important to have it over your nose as well as your mouth and to keep your hands out of your eyes. The infection route is through inhalation of aerosols or droplets that have virus particles on them and a little bit of hand to mouth if you touch a surface that's infected and then you put your hand in your mouth, for example. But it's not the most important transmission route. This is an airborne disease. And that's why untreated sewage is much more significant as a source of data than as a factor in public health risk. Infectious virions, so whole virions, have been shown to be pretty much absent in treated sewage and in river water containing sewage, and that's been demonstrated through experimental work. The viral RNA, however, is widespread in sewage and in water contaminated with sewage, and that helps us to monitor disease outbreaks. If we go on then to look at the presence of SARS-CoV-2 in water and wastewater and how it's removed, we're gathering a nice body of evidence. So the presence of severe acute respiratory syndrome coronavirus in water and wastewater has been reported widely. We know we can find it in water and wastewater. Bodily fluids of SARS-CoV-2 or COVID-19 patients and used masks, which are disposed of irresponsibly, are considered as the primary route of transmission of the virus into wastewater and into water bodies. Now, most coronaviruses which infect humans are inactivated very rapidly in water. 229E, one of the 4 endemic coronaviruses that I mentioned at the beginning, survives in water for 7 days. The RNA of SARS-CoV-2 was detected in untreated wastewater and secondary treated wastewater samples quite widely by Randazzo and his co-workers. However, the RNA of SARS-CoV-2 was not detected in tertiary effluent after the current disinfection process that they have in place with sodium hypochlorite alone. And under experimental conditions, it was found that the combination of sodium hypochlorite and ultraviolet light, use of peracetic acid, or high-intensity UV lamps all destroyed the SARS-CoV-2 virus in wastewater. This means that although current disinfection processes in tertiary stages of wastewater treatment plants killed totally the SARS-CoV-2 virus, the researchers didn't report the detailed conditions of the disinfection process. So, for example, they didn't provide the dosage of sodium hypochlorite or the contact time, and the effects of those conditions on the survival of all the coronaviruses. But it means that early indications are that standard tertiary treatment, standard disinfection, is adequate to remove the public health risk from SARS-CoV-2 and wastewater. In addition, the virions themselves are between 60 and 140 nanometers in diameter, and this means that membrane processes like ultrafiltration, nanofiltration, and RO will reject them. So we might need to think about disinfecting the reject from high-risk wastewater treatment works like hospital wastewater treatment works. Finally, there is an adsorption polymer out there which was already being designed prior to the pandemic for 2 other coronavirus targets. It had mixed success, but it would be very useful to find out whether or not this particular sorbent can also deal with SARS-CoV-2. To my knowledge, that work has not yet been done. So moving on to wastewater. If we look at what happens with SARS-CoV-2 in wastewater, we see that the wastewater generated by populations already infected with COVID-19, so COVID-19 patients, effectively hospital wastewater, generates much higher amounts of SARS-CoV-2 titer in the wastewater than you would expect just from the case numbers. SARS-CoV-2 is shed in stool and urine body fluids very early in the clinical course of the disease, and it infects also a large asymptomatic population, which makes it the ideal target for wastewater-based monitoring, which is what has developed over the last year. In the study on this slide, the authors developed a laboratory protocol to quantify the viral titers in sewage using PCR and polymerase chain reaction analysis, and they validated the results with sequencing analysis. They investigated the effects of things like rain events and diurnal or weekly flow variations on the amount of viral RNA detected in sewage samples. And the figures on the slide show the results of normalizing the data for sewage samples collected between the 18th of March and the 25th of March at 2 different sampling points. They are the north and south inlets at a big wastewater treatment works in Massachusetts in the US, so that's why it says north and south. On top, you can see the original SARS-CoV-2 titers averaged from the triplicate samples, and on the bottom, you can see the relative SARS-CoV-2 titers after adjusting for the dilution of raw sewage. And these graphs suggest that the number of positive cases estimated from the wastewater viral titers is an order of magnitude greater than the number of confirmed clinical cases, and therefore there's a significant impact on efforts to understand the case fatality rate and the progression of the disease. And this is one of the reasons why wastewater-based epidemiology has become so useful. The data help us to inform decisions surrounding the advancement or the scale back of things like social distancing and quarantine efforts based on dynamic wastewater catchment level estimates and estimations of prevalence. And that's what this slide now starts to talk about. In this study, which was a different group of researchers from the University Hospital in Milan and another 16 researchers from different collaborating organisations, had for the first time that we know of assessed the viral load of RNA and the amount of infectious SARS-CoV-2 virions in water. And it's a very important distinction to make because the viral RNA shows you that the virus is there or was there, but it doesn't show you whether or not the water is a medium of transmission. The presence of SARS-CoV-2 through its RNA has been demonstrated in 60 countries affected by this pandemic using RNA measurements. And the virus presence and infectivity in treated wastewaters and receiving water bodies are still relatively poorly investigated compared to that. In this particular study, raw and treated samples from 3 wastewater treatment plants and 3 river water samples in and around Milan were surveyed for SARS-CoV-2 RNA detection, again using real-time polymerase chain reaction or PCR, and an infectivity test on culture cells. SARS-CoV-2 RNA was detected in the raw wastewater, but not in the tertiary treated wastewater. They used 4 and 2 samples respectively on the 2 different dates. and the isolated virus genome was sequenced, and it belonged to the strain that was most spread in Europe at the time. The RNA presence in raw wastewater samples decreased after 8 days, which was interesting. It followed the epidemiological trend that was estimated for the area anyway. But the most important result that they generated was that virus infectivity was always null, and that indicates the natural decay of viral pathogenicity in time just from the emission. The samples of water from the receiving rivers were taken at 3 sites at the same time as the wastewaters, and they showed in some cases a positivity to real-time RT-PCR, probably due to untreated or badly treated wastewater effluent at the time, or to combined sewage overflows. But nevertheless, at the time they were detecting virus in the wastewater, they were still detecting infectivity of null. This is a very strong indication that the risks for public health should be very limited. Although these are only preliminary data, there haven't been many repeats of this kind of work yet, so we need to have a precautionary approach. But it means that the approach to risk assessment is just a precautionary approach, given the preliminary nature of the presented data, and the actual risks should genuinely be quite low. What this study showed us is that the virus enters wastewater treatment works. SARS-CoV-2 is deactivated, as I said, by many disinfectants in a matter of seconds, and because it's vulnerable to more disinfectants than E. coli, the indicator organism, we can assume that our tests hold true, that disinfection that removes E. coli has also removed SARS-CoV-2. Tertiary treatment can therefore be expected to remove SARS-CoV-2 to below the limit of detection, and that is so far what we've found. In wastewater treatment works, most of the viral material also appears to report to the primary solids. Now, this is going to have great benefits for us for monitoring efforts now and in the future, and I'll come back to that in a few slides' time. What it means immediately for staff working at wastewater treatment works is that the pre-existing guidelines that already were in place for protection of staff from all sorts of other viruses, including worse ones than SARS-CoV-2, to be honest, things like hemorrhagic fevers, other pathogenic coronaviruses, and then the waterborne viruses, which are much more dangerous in water than SARS-CoV-2, are still adequate during times of pandemic. So some of them have been updated. You can see a small list on the slide here. The most explicit one is the one on the bottom row, protecting wastewater professionals from COVID-19 and other biological hazards. But primarily, it's important to note that organisations like the World Health Organization, the Water Environment Federation, have made some updates to their guidelines, but they have not changed in their principles and working mechanisms. The activities we were taking before to make sure that staff are vaccinated against waterborne diseases, that they have gloves, that they have PPE, that they have eye goggles, that they have masks, are still valid and adequate for SARS-CoV-2. So if I can move away now from the staff themselves and have a little look back at a year of research on SARS-CoV-2 or COVID-19 transmission in water and wastewater. I mean, about a year ago, we were wiping down our groceries and worrying about touching doorbells and handrails. And it's hard to imagine now, but a year ago, the scientific community didn't know whether or not surfaces, droplets, aerosols were the most important route of transmission. We didn't know if it was a waterborne disease or not. So inevitably, here's a look back at what we've learned in the last year on SARS-CoV-2 and water and wastewater. So until now, there has not been one single case of transmission via contact with sewage or contaminated water. Nobody has caught COVID-19 from water. There are a few studies which have been conducted with aqueous matrices, and they have not detected infectious viruses, as I was just saying about river water. To my knowledge, there are only 2 studies so far addressing the infectivity of SARS-CoV-2 in raw and treated sewage and or natural water bodies. And this is Romaldi et al., the paper I presented a couple of slides ago, and Westhouse and colleagues who worked earlier this year on the infectivity of SARS-CoV-2 in sewage. Romaldi et al. Assessed the presence and infectivity in raw, secondary and tertiary sewage, and Westhouse investigated the presence of SARS-CoV-2 in raw and treated sewage and the infectivity in raw sewage from 9 municipal wastewater treatment works in North Rhine-Westphalia in Germany. They undertook flow-dependent composite sampling analyses, and although they detected viral RNA in all their samples, the same as Rimaldi, the results for infectivity were null in Germany, just as they were in Italy. And Westhouse et al. Concluded that sewage doesn't appear to be an important route of transmission. But they also emphasised the importance of carrying out further studies because they had a very small sample volume and a huge volume of wastewater. On the other hand, we have plenty of studies that have shown that SARS-CoV-2 can remain infectious for between 4 and 6 days in sewage or water respectively, and that other species of coronavirus may remain viable in these aqueous matrices for more than a year, depending on the sample conditions. Wang et al. In 2005 were working on SARS rather than SARS-CoV-2, but they were analysing wastewater specifically coming from the hospital of the Chinese People's Liberation Army. And what they discovered was that even after negative results for infectivity, they were able to suggest the sewage system was a possible transmission route because they demonstrated that virus survives and was culturable for between 2 and 14 days. And that means there are strong pieces of evidence that infection mediated by contact with sewage or contaminated water can't be ruled out yet, even though the preliminary data look quite good, because of other more resistant and infectious variants of SARS-CoV-2 that may appear in future. We already know that variants of concern are emerging at all times. So what this means is that SARS-CoV-2 can still be present in river water, that the public health risk is low, but the potential for using the presence of the virus in river water is very high for epidemiological studies in non-sewer communities. So perhaps not as relevant in the UK, but in parts of the world where there are peri-urban settlements, townships or favelas, and rural settlements that don't have sewage sanitation services, we can use the quality of urban rivers or surface water in place of sewer sampling for wastewater-based epidemiological work. I'm not going to dwell a great deal on this slide, but what it shows you is that the level of SARS-CoV-2 found in urban rivers where a city is only 3% of the sewage in Quito goes through sewage treatment works. Therefore, the urban rivers that flow through Quito can be used effectively to replace sewers when it comes to wastewater-based epidemiological work. And it means that the mechanisms of WBE analysis have broader application than in sewage treatment alone. So far, so good then. We've shown that SARS-CoV-2 can persist, or its RNA can persist into river water, even though the intact virions don't persist very well. They're dealt with very nicely by tertiary treatment. We've shown that even where virus particles are found in river water, they are not infective. We've shown that SARS-CoV-2 or COVID-19 are not waterborne. So this means that the public risk is relatively low when it comes to contact with water bodies. However, what does it mean for using river water in irrigation systems? If we're going to take that river water and spray it up into the atmosphere, what does it mean for the people nearby who might be inhaling aerosols from that spray irrigation? The transmission pathway from feces to water to foodborne transmission might yet be a transmission pathway for COVID-19. Sprinkler irrigation with wastewater and fertilization with wastewater solids, so with sludge, generates considerable aerosols and they can be inhaled. And this is important for farm workers. It's potentially relevant where agriculture takes place next to villages and irrigation and fertigation take place in relatively close proximity to suburban dwellings. It also indicates that transmission of disease can be minimized by disinfection prior to wastewater reuse. And this means we should probably only be irrigating with tertiary treated wastewater and only fertilizing fields with heat treated sludge. So that's what the implications are for public health in this setting. So if we turn our attention now to public health monitoring, we can look back at the early lessons from the first wastewater-based epidemiological studies that took place. We've already seen that there is no infectious SARS-CoV-2 in wastewater, or it's so low as to be a negligible risk. Multiple studies have not been able to culture SARS-CoV-2 from stool, despite high concentrations. Only 3 studies have managed it so far under very closely controlled laboratory conditions. It's going to sound like a strange choice of words, but the virus is quite fastidious when it comes to its conditions and nutrition. So is there a COVID-19 health risk from wastewater and water? Not very seriously, no. There are no epidemiological signals. Where we've had sudden outbreaks and super spreader events, they've all been around unmasked, high-dense populations in close proximity to each other, and it's been things like concerts and sporting events. Viral RNA in sewage is not infectious virus, and the SARS-CoV-2 shed in stool is itself not very infectious. It's quite difficult to keep it alive, in fact. No infectious SARS-CoV-2 has been detected in samples of water contaminated with sewage. What this means is that we can use the opportunities of that humongous piece of RNA that I showed you right on the very first slide. Environmental surveillance by testing wastewater for evidence of pathogens has a very long history of use in public health, particularly for other viruses like poliovirus, and more recently for things like antimicrobial resistance, or AMR. In the context of the ongoing COVID-19 pandemic, it's being used for the detection of SARS-CoV-2 shed into wastewater from bodily excretions, and efforts are ongoing to analyse historical wastewater samples for evidence of past SARS-CoV-2 circulation in countries like Italy, Germany and Brazil. This particular map comes from August 2020, so it's over a year old, and at that time you can see how many countries were investigating SARS-CoV-2 wastewater-based epidemiology, and how many were considering employing it as part of their national routine monitoring programme. If we can jump forward a year, the map looks very different. So this is the current information at WBE COVID-19, otherwise known as COVID Poops. It's a great website, and I'm sure you remember it. And it shows you that now the detection of SARS-CoV-2 through wastewater-based epidemiology has spread to nearly 60 countries. Nearly 3,000 sites around the world are using wastewater-based epidemiology. And even more than that, 265 universities around the world are assessing wastewater-based threats from SARS-CoV-2, and there are 95 dashboards available now covering multiple countries, including many in Europe, obviously the US and Asia Pacific. So far, only one in Africa, and that's where I am at the moment. Researchers from Yale University and the Connecticut Agricultural Experimental Station measured severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, RNA concentrations in primary sewage and sewage sludge in New Haven, Connecticut, during a COVID outbreak in spring of 2020. What they found was that RNA was detected throughout more than the 10-week study, and when they adjusted for time lags and they tracked the rise and fall of cases seen in COVID-19 clinical test results and hospital admissions, they showed that the RNA concentrations in the sludge were 0 to 2 days ahead of the positive test results by date of specimen collection, 4 days ahead of local hospital admissions, and between 6 and 8 days ahead of SARS-CoV-2 positive test results by the reporting date. The data therefore showed the utility of viral RNA monitoring in municipal wastewater as a 10-day early warning system for the population-wide level of COVID-19 cases. And this means in communities facing a delay between specimen collection and the reporting of test results, the immediate wastewater results can provide a considerable advance notice of the infection dynamics, about a 10-day gap between emergence of RNA in local sewage and accumulation of official reporting data. It's also possible to check the spread of variants of concern. Researchers at Ben-Gurion University in Israel have found a way to easily and cheaply get a much bigger picture view of the variant spread than it is with clinical case data by upgrading the coronavirus sewage monitoring programs that many countries already use. They've taken a standard test kit, which looks exactly like those currently used to assess RNA concentrations in sewage, but they tweaked it to screen for variants. They developed their tweak to the PCR test for the B117 or alpha variant, and then they made another version of the technology that works on the delta variant. And it's also likely that it can be tweaked to adapt for other mutations as they emerge. They've worked on one for the delta, for example. This is an absolute game changer in alerting authorities to the spread of variants of concern in the population and enabling them to take relevant steps for public health protection in the face of more transmissible variants. WBE currently provides a regional picture of coronavirus levels to health officials in parts of the US, the UK, Europe, Australia, South Africa and Israel, and the countries where those variants have emerged. But it doesn't currently tell them whether variants are on the rise, so this is an important tweak test. It simply uses new primers and probes to the Thermo Fisher test or the equivalent ones. Those are two key aspects of the testing systems, and it tailors them to new variants. So while it just is a tweak to the regular design of the test kit, the instructions of the kit users have not changed much, and that makes it easy to implement. What we can do with the wastewater-based epidemiological estimation is also to assess the effective reproductive number. Now, currently, the RE, or effective reproductive number, is assessed using clinical case data, and we use it to determine disease dynamics, to predict the spread of the disease and inform policies around local and national mask mandates, stay-at-home mandates, public health and protection measures, and estimate the effectiveness of those public health measures. And typically, we analyse it from clinical case data. However, using case data biases the RE estimate by things like changes in testing regimes and changes in reportings. So, importantly, the shift from testing in clinical settings to testing at home, where you can use a lateral flow test device at home, for example, is not picked up in clinical case data. So it's no longer a very good way of assessing RE. However, this study has showed that longitudinal measurements of RNA in wastewater can be used to estimate RE. Instead, it's been applied across different regions, and it provides an independent assessment of the dynamics of COVID-19. Given the widespread wastewater sampling that's taken place during the pandemic, these RE estimates are directly applicable as a rapid low-cost method to inform public health policy. The method can also be adapted to other pathogens, including ones for which we don't have clinical data available yet. And this means when there is the next pandemic or a new pandemic of another pathogen that we're not yet aware of, we'll be able to apply the same principles to the new pathogen. And this will make us much more able to respond more quickly when the next pandemic happens. Wastewater-based estimation of the RE of SARS-CoV-2 can be achieved using wastewater sludge rather than the wastewater itself. The lines on the graph on the slide at the moment indicate the RNA estimated using the RNA concentrations of three particular genes, the N, S, and ORF1A genes in blue, green, and yellow respectively in the wastewater, compared to the purple line, which is the RE estimated using confirmed cases in the area where this was done, Santa Clara County. The pale bands coloured in blue, green and yellow indicate the 95% confidence interval, and they indicate there was no significant difference because those green, blue and yellow bands completely overlay the purple band. This graph therefore shows that the RE estimates from wastewater are directly applicable as a rapid low-cost method to inform public health policy instead of using the clinical case numbers, which, as we've seen already, lag behind the RNA in wastewater or in sewage sludge by between 8 and 10 days. Now, it's important to note that sludge and sewage can both be used for wastewater-based epidemiology. Next, we're going to look at the benefits of using sludge compared to sewage. Now, do you remember when I had the slide of the wastewater treatment plant up that most of the virions reported to the sludge? Well, Graham and co-workers quantified the SARS-CoV-2 RNA in wastewater influent, primary settled solids, and effluent at 2 wastewater treatment plants to inform the pre-analytical and analytical approaches and to assess whether or not influent or solids harboured more viral targets and therefore represented a more sensitive case. The primary settled solid samples contain much higher SARS-CoV-2 detection frequencies than the corresponding influent samples. And likewise, the RNA itself is more readily detected in solids using one-step PCR than with two-step PCR, and this means you can get your results much quicker. It's probably because of reduced inhibition with a one-step PCR assay. Regardless of the cause, it means that we have a much more sensitive test if we undertake wastewater-based epidemiological studies using sludge than if we undertake them using sewage. And I'm going to show you a few graphs of what I mean. So Catherine and her co-workers subsequently analysed a longitudinal time series taking 89 samples from a single plant for RNA, and they also looked at coronavirus recovery techniques and fecal strength. For this, they undertook the studies using surrogate viruses, a bovine coronavirus, because it presents no threat to people, and a pepper mild mottle virus, which is actually a plant virus, but it behaves very much the same as SARS-CoV-2 and the other coronaviruses. What they found was that the confirmed new cases in the sewer shed of SARS-CoV-19, so the top graph in the top panel, and the 7-day smoothed new cases, which is the red line on the top graph, those represent the confirmed new cases of COVID-19. The middle and bottom graphs show you the concentration of the N1 and N2 genes measured in solids, so this is copies of RNA per gram of dry weight, and the error bars are standard deviations. The open symbols and non-detects are below the limit of determination, which is 40 copies per gram of dry weight per day. And if you take those graphs together, what they show you is that measuring SARS-CoV-2 RNA concentrations in settled solids is much more sensitive than measuring SARS-CoV-2 RNA concentrations in the influent. And this gives us an idea that if we undertake wastewater-based epidemiology using the solids and we report it immediately, it gives us, combining these results with the ones from Yale and Connecticut, a 10-day lead on what's likely to happen to clinical cases. Now, one of the complications of using wastewater-based epidemiology in suddenly 60 countries around the world is that not everybody is using the same methods. So we need to fill in some gaps in wastewater-based epidemiology. Comparing data around the world is very important for global learning. We need to understand what the results are from the study done in Italy, for example, compared to the one done in Singapore. In response to the COVID-19 pandemic, the international water research community very rapidly developed methods to quantify RNA in wastewater, and wastewater surveillance using the methods now, as you've seen, has expanded to 57 countries. However, as we all know, different research groups use different methodologies, and it makes it very difficult, if not impossible, to compare the results with confidence. So there were two studies. Zhao et al. Undertook a survey to find out how widely SARS-CoV-2 detection in wastewater is being investigated and what methods were used. And their results showed that the methods were rapidly applied to conduct wastewater-based epidemiology using things like primer probes. Around the same time, Pexon et al. In a different country worked out, or they sought to identify if and how the findings were impacted by laboratory methodological differences, things like sample concentration method, pre-treatments, primal probe selection, and solids removal steps. So in both studies, the aim was not to provide a standardized single method, but to evaluate whether the 36 existing methods that have been used around the world provide sufficient reliability and reproducibility to track trends in occurrence and assess the prevalence of community infection globally. In both cases, the researchers discovered that the methods were broadly comparable, which is very good news. It means there's a convergence of laboratory protocol development. They found that regardless of which method was applied, as long as the correct lab QC or QA measures were in place, you could use the trends to understand what was happening over time in a single place using the same method, or you could compare your data to data from another site where the results were generated using a different method and be 80% confident that your method would have produced the same data as the other site's reported results. What this means for those of us in practice outside of academia is that you can look at the results generated almost anywhere in the world using almost any method and be reasonably confident that if you'd done it at your site using the methods available to you, whatever they may be, you would have got within 80% of the similar results. So you can take other people's results and apply them to trend analysis on your own site. This means then that we're turning our attention to the implementation of wastewater-based epidemiology in a very big way. There are numerous resources available. A relatively recent development from the US Centers for Disease Control and Prevention, the CDC, and its effort to prop up the national wastewater-based surveillance system in the US to monitor the presence and trends of COVID-19 RNA in wastewater has provided a huge amount of practical guidance. And these are things like sampling strategies. So this is the nuts and bolts of how to develop a surveillance strategy if you don't have one, where to sample, how often, and what kinds of samples you need, how to store and ship samples to the laboratory for analysis. They've provided a range of testing methods, so sample processing, prep and concentration, the specifics of analysis and quantification, as well as what to use for controls and how to keep your samples safe. They've provided guidance on data reporting and analytics for the practitioner, including, these are specific to the US, but how to comply with health departments. However, there are useful parallels to be drawn around the world as well. Most importantly, they've looked at public health interpretation, so how to interpret the data that were generated, how to use those data in policy health communications, and what to tell the public if, for example, you're a water company serving a community, so you need to inform the community that you serve. And then there are some examples of what to do in particular cases like schools, universities, hospitals, prisons, and those kinds of things. The next things that will be coming out from the CDC are supplementary information resources, so utilities that have low resources, utilities that deal with very specific cases, specifically things like outbreaks in prisons, for example. But these are all very useful, practical, implementable strategies. There are numerous examples showing how wastewater-based epidemiology implementation has helped in different situations. So scientists in Hong Kong, for example, have combined RNA tracking in SARS-CoV-2 with source tracking, and they were able to have an early warning detection of an outbreak into apartment buildings. Finding SARS-CoV-2 RNA in the sewage led them to go in and test using nasal swabs all of the occupants of 2 different apartment buildings, and they detected previously undetected infections in some of the people living in 2 of the apartments. And by isolating those people, they were able to prevent the spread of COVID-19 into the rest of the apartment block. In California, the University of California has implemented wastewater-based epidemiology on its campuses as an early warning system. They have 343 buildings which are being monitored around campus to check for signs of RNA in the sewage, and whenever a sample comes back positive from any one of those buildings, the university sends messages to the students housed in that building to get themselves tested and to isolate until they get the results back. And what they've found is that they've caught up to 85% of cases on campus earlier than the development of symptoms in the COVID-19 sufferers. So they've been able to prevent numerous localised outbreaks on their campus. Now, this kind of level of wastewater-based epidemiology, where we go down to individual buildings and we can potentially track RNA of the virus, but also DNA in the stool samples or in the sewage samples back to individual occupants, has opened up a can of worms in terms of legal and ethical implications of wastewater-based monitoring of all sorts of things. Some government entities are considering whether to add community surveillance through wastewater monitoring routinely, so permanently, to assist in tracking disease prevalence and guiding public health responses, not only during the COVID-19 pandemic, but to emerging pathogens later on. However, with new technologies like the phone-based contact tracing that have been developed in response to the public health crisis, we've seen a pushback when it comes to civil liberties and protection of privacy. So wastewater-based epidemiology can potentially raise difficult legal and ethical questions. It's not a new topic, however, although it has revived the debate. Wastewater-based epidemiology has been used extensively in the past for tracing things like illegal drugs in communities. Using it to monitor illicit drug use has been mentioned during interviews with Bloom and Ultra, for example, who are undertaking wastewater-based epidemiology now on behalf of the Canadian government. And in the Netherlands, there is already widespread wastewater analysis to monitor illicit drug use. The reason the legal and ethical implications are not too severe is that our wastewater is mixed up with wastewater from several other people. In the Netherlands specifically, there is no ethical concern in populations with over 10,000 inhabitants, but the researchers need to minimise the possible adverse consequences of studying smaller populations. So we're unlikely at this point to start applying wastewater-based epidemiology at household level. For things like apartment blocks or housing units like the University of California, it's absolutely fine. When it comes to looking at the wastewater emanating from drains from individual homes or buildings, there might be another debate to be had. And for example, this is exactly what the Australian CSIRO is proposing to do. So we're waiting to find out what happens with the ethical debate in Australia. One of the arguments against water-based epidemiology in some circles has been the cost. And in response, Hart and Haldon did a nice study on the economy and opportunities and challenges for and against using wastewater-based epidemiology. They examined wastewater computationally as a matrix for detection of SARS-CoV-2, and they found that one infected individual theoretically is detectable among between 100 and 2 million people. This means that WBE is a way of monitoring 2.1 billion people globally by simply applying WBE to about 105,000 sewage treatment plants. And this means it's a much more cost-effective option than clinical testing of 2.1 billion people. So for resource-poor regions, well, for anybody, wastewater-based epidemiology might represent the only viable means of effective surveillance of a whole population, where we can't realistically ask people to test themselves once or maybe 2 or 3 times a week. I will wrap up here. I'd like to say thank you for your attention. Any questions or comments will be more than welcome.
Hello everybody, thank you very much for listening.
And if you would like any more information, we are doing
have
our
some
next
questions
Water
that are
Action
coming
Platform
in.
webinar in a few days' time on the 28th of October, and I might see you there too. Thank you very much.
The first question, when HIV first became a concern for workers in the water industry, particularly when the issuing of needles to drug users by public health teams led them to being flushed down the wastewater system to form huge porcupines in the Copa sacks at wastewater treatment works, water companies had to undertake relative hygiene awareness training for their employees and contractors very rapidly. Is there likely to be the same need for SARS-CoV-2 related hygiene awareness training?
Great question, thank you. The short answer is that it's no. That's the really short answer. The slightly longer answer is kind of yes, to be honest, because the route of transmission, or the route of infection rather, is through the respiratory system, so eyes, nose, mouth. As long as wastewater treatment workers are using masks, face shields, eye goggles, gloves, not putting dirty hands in their mouths, for example, then there is no need for anything in addition when it comes to protection from potential infection from the wastewater itself. However, because the wastewater is not the only source of infection, there is a need to enforce or require other precautions on site, and that's because workers can infect each other. So it's not a case of adjusting the the SOPs around biological hazard handling from the sewage, but we need to maintain things like social distancing, keep people 2 meters or 6 feet apart on site, stop them from unmasking in unventilated spaces, those kind of precautions, but exactly the kind that we've had in public spaces and restaurants and shopping malls and public situations. So if people are wearing masks in public places or in enclosed spaces at work, they're maintaining their social distance, they're not picking up a pen that everybody in the tea room has used and putting it in their mouth, then they should basically be fine. It's not particularly different from general public health protection and your normal biohazard protection at work.
And then a second question, also related to workers or people on wastewater treatment works. I mean, some of the processes we use generate aerosols. For instance, spray generated from surface aeration systems. Is that likely to be cause an additional hazard? Is that something that workers need to be a little bit more aware of? For instance, which way the wind's blowing when they walk past the equipment?
Yeah, it is possible. They do need to be aware of it. It's a little bit debatable because infective virus hasn't yet been isolated from wastewater, but we should take a precautionary approach and assume that SARS-CoV-2, like any other common viral pathogen or bacterial pathogen found in wastewater, could survive in aerosols when wastewater is mixed and aerated. So we need to take precautionary measures like good ventilation, limited crowding. If you're working at an outdoor site, that's probably not too difficult to do. It's a little bit complicated because droplet transmission, so that when I say droplets, I mean about 5 micrometers in diameter. Droplet transmission is thought to be the major mode of transmission. And aerosols, which are less than 5 micrometers in diameter, have been very difficult to sample, and it's been very difficult to sample and characterize the viability of airborne viruses in aerosols, largely because there are huge technical challenges with doing things like sampling aerosols and not changing the nature of the aerosol through the sampling process. In the laboratory, it's been demonstrated that SARS-CoV-1 in air and SARS-CoV-2 in air can survive in droplets and aerosols for up to 16 hours. However, outside the laboratory, the results are much more contradictory. There have been a few studies, none of them have been particularly conclusive, but it hasn't been possible to either rule out exposure and transmission from aerosols, but it hasn't been possible to prove that it's ever happened either. So there's no evidence yet that anybody has caught COVID-19 through aerosol transmission to wastewater treatment works, but it hasn't been possible to rule out that could potentially happen. So again, similar to the other pathogens, if you are working around aerated wastewater that hasn't gone through tertiary treatment yet, then you need to be using masks, using gloves, using eye goggles, using face shields, and taking care not to inhale a large amount of droplets or aerosols around site. Not because it's been shown that that is definitely an infection route, but because we have not shown that it isn't.
Right. Slight change of topic. How and where do you see the lessons learned from the SARS-CoV-2 experience and water-based epidemiology being used in the future?
I think we will be able to deploy the methodologies much quicker when the next pandemic happens. We know that it will happen. People in the medical science research arena have been saying we've been overdue for a pandemic for a few years and that we're not equipped to deal with one, and it turns out they were absolutely right. The wastewater-based epidemiology principles aren't new, and the country where I'm sitting right now as I talk to you, South Africa, already had a WBE program in place because we have much worse viruses like Ebola, for example, right up to the borders between ourselves and our neighbours to the north. So we've got WBE in place for other pathogens, including viruses. We have an active WBE program for poliovirus. Until now, it's depended on the detection and characterization of the pathogen. But something that's been new knowledge that's been generated in the last year is the ability to use WBE principles without the need to identify the pathogen. And that means that we'd be able to start monitoring programs to protect public health and predict outbreaks before needing to characterize the disease up front. So I think that will be the big lesson learned from this particular pandemic. In the case of future pandemics, as soon as we've got a fragment of RNA that we can detect and say, this is the culprit, we'll be able to put WBE monitoring in place much faster rather than going, well, what does it lead to? What disease does it cause? What are the clinical outcomes? All those other sort of, what are the signs and symptoms questions that we felt we needed to answer sort of this time last year. So I hope that will be the big lesson learned. If something's going to come out of the calamity that was good for humankind, then I hope that's what it is.
Thank you very much, Jo. Thank you very much for your time. We're about out of time now. Just to remind people, there's a link shown on the screen where people can listen into further developments of this on the Water Action platform that Jo participates in. The next one's happening on Thursday, the 28th of October. It's on twice during the day at 7:30 in the morning and 4:30 in the evening for us in the UK. It just remains me to thank you, Jo, for your time today. A very interesting webinar, and thank you very much. And thanks to everybody for listening in.
My pleasure. Hopefully see you on Thursday.
See you on Thursday. Thank you.